Hi guys, so this video is going to go a little bit more in depth into genetics. We're going to start looking at genetics like Mendel never really did. All of Mendel's work pointed to DNA and genes and traits being inherited singly, or even, you could inherit two at a time, but you always inherited one and then the other, and then one and then the other. And that's not always true, because early Mendel genetics makes it look like you're either dominant or you're recessive, and that's the only thing you can be. There's no way to form a blending of genes, to use a descriptive term, or there's no alternative, and that's not true. So that's what this video is going to show. It's going to show when genetics goes extreme, for lack of a better term. So here we go. So, like I just said, Mendel's system was fairly simple. If you think of what he worked with peas. Those peas were genetically simple organisms. They have a couple of traits that stand out, and those traits have the ability to be controlled by just a single gene. Each gene has two alleles, and you get one from mom and one from dad, and one gene is completely dominant to the other. But that's not how it really works. It's rare that genotype and phenotype are that simple and that basic. So the first kind of situation or scenario that we will look at is something called incomplete dominance. So far we've learned that one gene's dominant and it overpowers or suppresses or is expressed in favor of the gene that is recessive. So for example, with Mendel and his the color of his pea flowers, one was purple, one was white. Purple's always dominant, so purple's always shown, even when you have a hybrid. That's not always true. In the case of incomplete dominance, the heterozygote, the actual hybrid, shows a different color. It's a blended phenotype. So if you look at roses, for example, or maybe even carnations, I don't know. I know roses do this for sure. You can have a red rose and a white rose, but it is also possible to have pink roses. Pink roses exist because of incomplete dominance. So again, incomplete dominance is where the heterozygous condition has its own color. That color is a combination of your dominant phenotypic color and your recessive phenotypic color. Excuse me. All right, so let's look at it. So we can take a true breeding red flower and cross it with a true breeding white flower. Technically, it would look like red would be dominant to white, but the resulting F1 generation is all going to be pink flowers. We allow those pink flowers to self-pollinate, and look what happens. We get a 1 to 2 to 1 ratio of red to pink to white. So no longer does the red color fully dominate, or no longer does the dominant allele fully cover up the recessive allele. Now they actually blend together, and you get a completely different phenotype from either parent. All right, so that's one example. Now another example of how genes can be a little bit more complex or complicated would be through codominance. So in codominance, you have two alleles, and they both affect genotype equally, but separately. I know, that's weird. So let's take a minute to wrap our minds around that. We have two alleles. Both those alleles affect the genotype, but they affect them at the same amount, but separately. So in this scenario, there is no blending. You don't get a pink flower. A good example of something that exhibits codominance is the blood groups or the blood types of humans. We use what we call the ABO system. So there are three alleles when it comes to blood. There is, now we use I because I stands for immunoglobulin, which is the protein responsible for blood. So IA is one of those alleles, IB is the other, and the recessive trait in this case is showcased by lowercase I. I want you to notice that both A and B are capital or uppercase. Normally we associate uppercase with being dominant, lowercase with being recessive. Well, it's still true. So why do we have two different letters that are both uppercase? These are not homozygous. Well, it's because the A allele and the B alleles are both co-dominant. They both affect the blood type, but they do so separately. Excuse me. So your blood type is generated, by certain proteins that you find on your actual red blood cells, so on the surface of your red blood cells. People with type AB blood actually exhibit both the A and the B antigen. Their blood cells have both of these proteins on them. One's not more dominant than the other. They coexist and they both affect the phenotype. The only one that's recessive would be I. And if you have two of them, then you have O blood. So if we look at this chart, I'm going to try to write in as best as I can what these things are going to look like. So there are four phenotypes for when it comes to blood. Type A, type B, type AB, and type O. But A and B can have two different genotypes. You can be homozygous dominant for the A blood type, or you could be heterozygous for the A blood type. So the genotype of someone who is type A and homozygous dominant would look like this. I and capital A and I and capital A. Or if you're heterozygous, you would be I and capital A and lowercase I. All right? So what does it mean? It means that on your blood cell, you produce or you make A antigens. Now if you're B blood type, it's kind of the same thing. You can be I and capital B and I and capital B, or you could be I and capital B and lowercase I. If you are B blood type, it means you make B antigens on your red blood cells. Now, the exception to the rules would be AB. AB has only one genotype. You are sharing dominance. So you are I in capital A and I in capital B. You have to have both in order to express AB blood. If you are O, you are the recessive, in terms of on your red blood cells, well, you make both antigens. You make antigen A and antigen B. If you are O blood, you are the recessive trait. So you are lowercase I and lowercase I. So what antigens do you have? None. You don't make any. All right. If you want me to go over the antibodies and the donation status, I can do that in class for you. So we're going to move on. All right, so here comes another condition, pleiotropy. A lot of genes are pleiotropic, which means that one gene actually affects more than one phenotypic characteristic. It affects more than one trait. So for example, we have conditions like dwarfism and giantism. That would be an example of conditions that are pleiotrophic, where you have one gene, but that one gene affects several different traits or features or characteristics. Right, so here are some famous examples of people that suffer with giantism, like Andre the Giant, big wrestler guy. And then examples of, if you watch the show Little People, Big World, I think it's called, they suffer with this particular type of dwarfism. And when I say suffer, I don't mean that it's a burden, but that's the condition they have. So let's go through and look. This is a type of dwarfism. It's one of the main ones. And it's the result, I'm sorry, this came up wrong, but we'll talk about this in a minute. It's the result of a heterozygous condition. So it's what we call a dominant inheritance. If you inherit, if you're heterozygous for the trait, then you will have that dwarf condition. If you are recessive for the trait, you will not have that dwarf condition. On the flip side, if you are homozygous dominant for the trait, that actual gene is lethal and the organism will not survive. So if we look at the crosses, if you cross someone who is heterozygous for achondroplasia, I can never say that word, but anyway, with someone who is recessive for that same trait, there is a 50% chance that offspring that they have will have this condition, this dwarf condition, and 50% of there's also a 50% chance that their offspring will be normal in terms of height. But if we cross two people who are heterozygous for this dwarfism trait, then there is a 25% chance that their offspring could have the lethal combination of that gene. In this case, that lethal combination is homozygous dominant. The homozygous dominant condition results in death, for lack of a better term. Then there is a 50% chance that they could be a dwarf, they could have that condition, and a 25% chance that they would be normal when it comes to that particular condition or gene. Now, another condition that we can look at is the condition of epistasis. In epistasis, one gene completely masks another gene. A good example of this is coat color when it comes to animals like mice or even Labradors, like certain types of dogs. Their coat color is controlled by two separate genes. They have a gene that determines whether or not they will be pigmented or not pigmented, and then they have another gene that's going to determine the actual color of pigment that they can express. So the C allele controls pigment versus non-pigment. If you are dominant, you will be pigmented. If you are recessive, you'll have no pigment. And then the B allele controls whether you express a black color or a brown color. Big B, black color, little b, brown color. So that means that if you end up with being recessive for pigment, you are going to be albino. It doesn't matter if you have a big B or not. This recessive condition trumps this condition. So instead of having our nice 9:3 to 3:1 ratio, the ratio becomes a 9:3 to 4 ratio. If you look at the picture, I'm going to try and show you. So remember, the grand master in this particular example is this C gene. If you have two little c's, automatically you are not going to have any color. It doesn't matter what your B allele looks like. So these mice here are all at least heterozygous for both C and B, these mice that are colored, right? So that's why they're all black. Versus the mice that are brown, they are recessive for color. Well, they're recessive for pigment, so that's why they're brown, but they're all at least heterozygous for color. So they get to be colored because they have at least one big C, but they can only be brown because they are recessive for the pigment color. Now, these three mice all have at least a dominant allele for pigment. They have at least one big B. So that makes you think that they should all be black. But no, if you look again, you'd see that they're all recessive for having pigment. So they're all albino, no pigment at all. This last little mouse here is recessive for everything. He's recessive for having pigment. He's also recessive for the color of pigment he has, so he's also white. So hence the 9:3 to 4 ratio. 9, let me highlight these for you. 9 to 3 to 4. So you're probably thinking, well, that could have just been by accident. How do we know that this is going to happen? Well, in biology, I know we don't do a ton of math, but we do some math, and most of it is for proofs. So what we use to prove that this isn't luck or random chance is a test called the chi-square test. And one of our labs will deal with you learning how to use the chi-square test. Okay, so here's epistasis again, but this time it shows in dogs. So again, it's two genes, E and B. The E allele codes for whether they get to be pigmented or not pigmented, and the B codes for whether they will be black or brown. So what we call a yellow lab just actually means that they don't have a dominant E. They're completely recessive for E. And it works out that they don't have actual dark pigmentation in their fur, so they end up being kind of yellow. Okay? And there are different varieties. Like you can have a yellow lab that has brown features, like its nose is brown, its lips are brown. But you can also have a yellow lab that has black features. And then in terms of the chocolate lab, another one of the non-Mendel-like conditions is called polygenic inheritance. So poly means many, genic for gene, so many gene, or many genes, sorry. So when you think about it, what this really is when a phenotype is determined by the additive effects of two or more genes on one particular trait or characteristic. So it's like these two separate things are affecting one phenotypic trait. We have a lot of human traits or human characteristics that fall under that umbrella term of polygenic inheritance. So things like our skin color and our height and weight and intelligence, and even some kinds of behaviors. If we just look at skin color as an overall example, if you are Caucasian, you are fair skinned. But you can look at someone who is American but Caucasian, English but Caucasian, Scottish but Caucasian, and you'll see different variations in the actual color, tone, appearance, whatever, of their skin. So even though they're all the same ethnicity, they don't actually look the same. And yes, environmental conditions do play a role in skin color, but inherently it's also affected by these polygenic traits. All right, so let's look a little more closely. Albinism is a good example of how, regardless of your ethnicity, your skin color may not reflect your ethnicity at all. So albinism, I can never see that word for some reason, can be inherited as a single gene trait. It is recessive, so in order for you to have it, you have to have both of the letters, and it's where your body does not produce the universal pigment that we all have, which is melanin. Now, if you're fair-skinned, you just have a small amount of melanin, but if you are darker skinned, you have more. When you are albino, your body doesn't make it at all, because one of the precursors to melanin, which is the pigment that makes you colored, that gives you a color, one of the precursors, like I was saying, is tyrosine. Well, if the enzyme that is supposed to convert tyrosine into melanin malfunctions, then you end up with the condition of being albino. Now, there's a difference between being very light-skinned, but your hair will still have pigment, your eyes will still have pigment, your eyelashes, your eyebrows, you'd still be pigmented in certain places. When you are albino, you don't have pigment at all, so you end up with this very blonde-looking appearance to hair, eyebrows, eyelashes. Your eyes tend to be yellowish or reddish, just depending. And it has nothing to do with your actual ethnicity. So there are people of African descent who are albino. It's very interesting. All right, so here are just some pictures. These pictures just showcase some of those features I was talking about. Like if you look at the eyes right here, you'd see that they're red as opposed to being blue or brown or green or hazel. Look at the very light colored hair. That's very characteristic of people with this particular condition. Okay, another condition or another series of conditions that do not follow Mendel's original plan come through what we call sex-linked traits or sex-linked characteristics, where the sex of the baby, meaning is the baby a boy or a girl, or the offspring a boy or a girl, it carries certain genes. So the genes are found on the actual sex chromosomes, on the Y chromosome or on the X chromosome. And they're not going to be autosomal characteristics because they're not found on autosomal chromosomes. All right, so just to remind you, all of these here would be our autosomal chromosomes, this whole region here. Whereas here, these are our sex chromosomes. So the traits and the characteristics we're about to talk about are only found on the X or Y chromosomes. So the scientist who discovered sex linkage was a guy named Morgan. Now, ironically enough, he worked with Drosophila, or fruit flies. So for the duration of time, whenever we talk about how sex linkage was discovered, we always refer to these flies as Morgan's flies. So you probably will hear me use that term in the next couple of slides. So he chose Drosophila for a couple of reasons. They're cheap and they're easy to find, and they have a distinctive XX female and XY male, and there are a couple of different traits that you can attach to those chromosomes and breed them for. So what he did was he took a true breeding red-eyed female and mated it with a true breeding white-eyed female. The result were 100% red-eyed offspring. So both the girls, sorry, not both the girls, but the girl flies had red eyes and the boy flies had red eyes as well. So then he let them mate, and the result for the F2 generation were all the girls had red eyes, but half of the boys had red eyes and the other half had white eyes. So if we go back to the F1 generation, we knew that these flies would have been hybrids. They're heterozygous because their parents were red-eyed and white-eyed. When we look at the F2 generation, all of the girls are still red-eyed. It's only the boys that seem to have this white eye phenomenon in this F2 generation. So this separation of eye color based on sex tells us that the sex of the individual does matter when it comes to this particular trait in flies. Okay, so let's see if we can figure out why we saw the discrepancy that we did with Morgan's flies. Now, these two Punnett squares represent the traits of eye color without taking the effects of sex into play. So we're looking at crossing just the red-eyed phenotype with white-eyed phenotype, and we would get 4 heterozygous or 100% red eyes, just like we did with Morgan's flies. But when you take your 2 heterozygotes and you cross them, it looks like we should get 3 red eyes and 1 white eye, and that the genotype should be homozygous dominant, 2 heterozygous, and homozygous recessive. But that's not how it works, because when we did these crosses, we did take into account sex, boy versus girl, and we saw a difference there. So it means that when we do our Punnett squares, we need to take into effect the sex as well. In both humans and mammals, there are 2 sex chromosomes, X and Y. If you have 2 X's, you are a female. There is gene redundancy, just like there is in all of your other autosomal chromosomes. Gene redundancy just means that you have 2 copies of a particular gene. If you are X and Y, then you're going to be a boy. There is no gene redundancy because the traits on the X are not coded for on the Y. They're not homologous, this X and Y chromosome. They're not homologous. So you become a girl when your mom passes on an X and your dad passes on his 1X. You become a boy when you get an X from your mom, but your dad passes on his Y. So only the males of the species can control whether or not the offspring are male or female. It's not like they can inherently control that, but they're the ones that pass on the determining chromosome. So now let's look at Morgan's flies, taking into consideration the sex of the flies. So when you look at the female fly, we're going to be XX and we're going to be big R because it was a true breeding female fly. When we look at the male fly, we're going to be X and small r, and note that the Y has no small r because the eye color gene in this scenario is only found on the X. So whatever is on this X, that's the type of eyes that that particular fly has. If it would have been a big R, then that fly would have had red eyes and not white eyes. This is the only time where you can have the recessive trait and you don't actually have both alleles. You only have one because males only have one X. So when we cross them, we end up with 100% red eyes. All of the females are red, and all of the males are also red. So when we take our hybrids and we cross them, this is what we're going to get. Now here's what you have to remember. For the boys to be red eyes, their X has to have a big R on it, okay? Whereas for the girls, they could be red-eyed with 2 big R's or 1 big R, 1 little r. So when we cross them, we're going to end up with 100% red eyes for females and 50% red eyes for male and 50% white eyes for male. So once we put the sex chromosomes into place, then Morgan's discoveries made sense. But when we were trying to do this without taking into consideration the sex, it didn't make any kind of sense. We would have seen what Mendel would have expected us to see. Okay, so what genes are on this Y chromosome? Well, it doesn't have a ton of genes on it. It does have what we call the SRY, or sex determining region, which is the master regulator for maleness. It's what inherently makes a boy a boy. It turns on the genes for the production of those male hormones. And this is another example of pleiotropy, where you have many different effects from this one region of your chromosome. Now, on an X chromosome, you're going to have your sex-determining things other than your sex-determining region, so lots of other genes and traits. Some classic things that can be affected on the X chromosome are conditions like hemophilia and muscular dystrophy and color blindness. Now, if you're a girl, it may not be an issue because if you have because you have 2 X chromosomes, while one X might have defective genes on it, the other X may have genes that work perfectly fine. So you yourself, you don't exhibit traits of the actual condition. But if you're a boy and something is wrong with the genes on your X, you have the condition. There's no other alternative because you only have that one copy of that particular gene. Okay, so this is just to give you an idea of some of the different things coded for by the human X chromosome. There are lots of conditions, as you can see, some of which you may be familiar with and some of which you may not be familiar with. But there are about 60 diseases that are all traced to genes on your X chromosome. And they're X-linked because they are attached specifically to just the X chromosome. So if you're a girl, like I said before, this may not be such a bad thing. But as a boy, it could be a bad thing. All right, so this is the slide you've been asking me about, or I've been hinting about for a while. Now, I told you that for the most part, the Y chromosome only codes for the sex-determining region, and then it doesn't have a whole lot more on it. So this is just my take, my attempt to be funny. I hope you enjoy it. So I'll give you a little while that you can read this and hopefully have a laugh. All right, we're going to move on. So one of the classic examples of an X-linked or sex-linked condition that has plagued the royal family of Europe for generations is the case of hemophilia. Hemophilia is a condition where the person does not produce or lacks the ability to clot. So if they get a wound, they'll bleed out unless, they seek immediate medical attention. It's famous because of Queen Victoria, who, if you look at our little pedigree here, let me see if I can highlight for you. So here's Queen Victoria. Now note, above Queen Victoria, all of the people she is descended from, none of these people have this hemophilic condition. But she is a carrier of the condition. So at first it doesn't seem like such a big deal, but she does have a daughter, and her daughter is also a carrier of that hemophilia condition. On this side of the family, she had another daughter named Victoria who got married to some guy named Frederick, and they made up the German royal household. Note, they don't have any traces of hemophilia, not even carriers. So the German royal house doesn't have this condition. Everybody else kind of does. The British royal house and Spanish and all that. So if you just look everywhere you see that half shaded in region, that tells you that that woman is a carrier of the condition hemophilia. Wherever you see a fully shaded in square, it tells you that that man has hemophilia. And this picture is in the way, but in class I can show you the slide again, take this picture off, and we can really dissect this pedigree in more detail. So, what's hemophilia? Well, I already told you what the disease is, like, some of the symptoms, the fact that you don't clot. But for genetic purposes, hemophilia is a sex-linked recessive trait. So, let's go through and look at it. Now, girls have three possible scenarios. Either they're fully healthy, they're carriers of the condition, or they actually exhibit traits of the condition, actual signs and symptoms. Most often, females are carriers, and it's because we have two X chromosomes. So even if one X chromosome has malfunctioned, the other is normally okay. So it's a situation where if you are homozygous dominant, so capital H, or you are heterozygous, capital H, lowercase h, you don't actually have the condition. The only way you can have the condition is if you are lowercase h, if you're a girl. But because boys only have one X chromosome, if they get the messed up or the malfunctioning X chromosome, the one with the little h on it from their moms, then they're going to automatically have the disease because they have nothing else to balance it out. They cannot be heterozygous. So with these sex-linked traits, what you have to realize is that males cannot be heterozygous or carriers of any of these diseases actually, whereas women can. Okay, so I hope that makes sense. So in this scenario, she'd be fine, he'd be fine. She would be a carrier, but ultimately she'd be fine. He would have the disease. So not fine. Another condition that is sex-linked, and this time affects females, we see it in female mammals, is something called X inactivation. So female mammals inherit two X chromosomes. In X inactivation, one of those X chromosomes becomes inactivated during embryonic development. So as that embryo is developing, as the fetus is actually becoming a fetus, one of the X chromosomes is inactivated. It's like shut down or turned off. What it's going to do is it's going to condense into a compact object that we call a bar body. Which X becomes inactivated is completely random, but the result is a change in the phenotype. The change in phenotype that you would see would be a color change. And a good example of these would be in what we call calico cats, the ones that are mostly white, for example, with patches of like brown or orangey tones and like black. Those colors are caused by the inactivation of one of those X chromosomes. So you get this patchwork, mosaic-like looking phenotype. All right, another sex-linked trait is male pattern baldness. It is an autosomal trait that's influenced by sex hormones, and it normally has an age factor. It tends to set in after the individual is 30 years of age. It is dominant in males and recessive in females. So if you have the dominant trait, you're bald if you're a guy. The only way you can be bald and be a girl is to get the recessive trait. So that means you have to have two recessive alleles, little b, little b. But in guys, because it's dominant, as long as you have one big B, you'll be bald. So that means homozygous dominant, you'll be bald. Heterozygous, you'll also be bald. I just thought that this particular picture was funny. Okay, now, other than the actual genes, your phenotype can be controlled by your environment. So sometimes the phenotype you see is as a result of the initial behavior of genes or the initial result of genes, but it can change over time due to the environment that you are subjected to or that you live in. So if you think of color, like the color of flowers, that could be an example. Hydrangea flowers are characteristically blue or pink, just depending on if they're grown in soils that have low pHs or high pHs. The effects of temperature and coldness, even the change of background in our environment, can also influence our colors. If you think of animals that live in the Arctic, especially if they're mammals, during the winter time, their fur or their coat blends into that background. So like this Arctic fox right here, who's so cute. Anyway, when it's snowy out, it's going to be a white background. Well, normally these are darker colored animals. If you stick this darker fur on this animal, he's going to stick out like a sore thumb on this fully white background. Polar bears and anything else that's going to be a predator of his can easily find him and pick him off. So in the wintertime, they shed all of their colored fur and they keep this white lawn of color so that they blend in. But in the summertime, where you have grasses, some of which are brown and green, or they're changing color a little bit, there is more color in the background, then the Arctic fox is going to change its coat as well into this more blended shade of browns, like dark browns and light browns, and even some white, so that he blends in to his background more easily. And his ability to change this really is dependent on temperature. It's more of a heat-sensitive thing. Even when you look at us and our skin tones, if you are found in areas that are further away from the equator, or you stay inside a lot, or you live in a place that's really cold, you tend to be of a lighter complexion than if you go outside all the time or you live close to the equator. Now, it doesn't mean that you can go from being really light-skinned to being really, really dark-skinned, but if you think of your skin tone in the summertime versus the wintertime, there is a difference. Now, boys may not get this as much, but girls should probably get this. For those of us who wear makeup, we normally have two different shades of makeup. One for our summertime skin, which tends to be darker, and then one for our wintertime skin, which tends to be a little bit lighter. Okay, so I hope the video was fairly clear. I know we will go over the dwarfism and giantism traits. Like, it's one of the labs that we'll do this week coming up. So if you didn't get that part as well, don't stress too much. Just make sure the other stuff is stuff that you understand. Make sure you're coming with your questions on Monday and Tuesday. See you guys soon. Enjoy your weekend. Bye.